Welcome to the third part of our symposium. And it is my pleasure to introduce Cristina Firincă with the talk Bioremediation Strategy for Chromium Contaminated Soils. Cristina, please.
Good afternoon. My name is Cristina Firincă. I am part of the microbiology laboratory from the biotechnology department in NeXT-Chem, and today I will present the results of our research in regards to a bioremediation strategy for chromium-contaminated soils. Chromium pollution can have various sources, such as smelting, mining, electroplating, leather processing, utilization of pesticides and other agricultural products in agriculture, and even in cigarettes. Each gram of tobacco can contain up to 2000 nanograms of chromium. In the European Union, there are regulations for the total concentration of chromium in agricultural soils at 200 milligrams per kilogram and in drinking water at 50 micrograms per liter. Chromium can be found in two stable forms. Trivalent chromium has physiological utility, but can be toxic in high doses, and it's generally less bioavailable to living organisms. Whereas hexavalent chromium is highly toxic even in low doses, producing mutations and various forms of cancer. And it's generally easily uptaken by living organisms. For this reason, numerous remediation methods are available. Physical and chemical methods can require significant costs and utilization of resources, can affect soil structure and fertility, and can generate the production of large amounts of waste. On the other hand, biological methods can be more economic, more efficient, and have less of an impact on all forms of life and the environment in general. The aim of this research was to evaluate the level of contamination with chromium in soil and isolate chromium-resistant filamentous fungi for future applications in bioremediation. For this purpose, we have analyzed the physicochemical and biological parameters of the contaminated soil. We have isolated filamentous fungi and tested their resistance to different concentrations of chromium. Soil samples were collected in February of this year in Bucharest, and the physicochemical parameters that were analyzed were pH, moisture, organic matter content, and total concentration of chromium. Basal respiration and substrate-induced respiration were quantified by titration with hydrochloric acid. The microbial biomass was extracted through fumigation with chloroform, and based on these results, the metabolic quotient was calculated, representing the carbon dioxide produced per unit of biomass. Low values of this quotient indicate a low level of stress exerted on the soil microbiome, as well as an efficient use of the carbon from the substrate. Filamentous fungi were isolated through serial dilutions distributed on agar media supplemented with a low concentration of potassium bichromate and chloramphenicol. And their tolerance was tested for concentrations between 50 and 1000 milligrams per liter of potassium bichromate. Values above 0.8 of this index show a high level of tolerance to chromium. Additionally, microscopic and macroscopic observations were carried out for a better assessment of the tolerance level. Next, I will present the results we have obtained through our research. The soil was characterized by low moisture and organic matter content, as well as a slightly alkaline pH. In correlation with the significantly high concentration of chromium we have detected, these conditions can exert a level of stress on the soil microbiome. The presence of this stress was confirmed by the low levels of basal respiration and substrate-induced respiration. The total concentration of chromium was positively correlated with the basal respiration and negatively correlated with the substrate-induced respiration, indicating an active metabolism focused on survival instead of growth and development. The microbial biomass registered high values, also positively correlated with the total concentration of chromium. And in association with the low metabolic quotient, this can indicate the presence of metal-tolerant microorganisms in the contaminated soil. In that sense, we have successfully isolated 19 filamentous fungi belonging to the genera Trichoderma, Penicillium, Fusarium, and Aspergillus, genera that have been proven to studies to have bioremediation potential in heavy metal contaminated soils. In regards to the tolerance to chromium, we have observed that at the lowest concentration of potassium bichromate, almost half of the strains displayed high tolerance, whereas the other half displayed very high tolerance. An increase in the concentration of potassium bichromate was correlated with a decrease in tolerance, more significant, starting with 250 milligrams per liter. At the highest concentration tested, only 3 strains presented moderate tolerance, whereas 1 strain was generally highly tolerant to all concentrations tested. In regards to the growth rate, we have observed that low concentrations of potassium bichromate determined stimulation in growth, and an increase in concentration was correlated with a constant decrease in growth rate up to a point of complete inhibition at higher concentrations. The exception was 1 strain for which no significant differences could be observed in terms of growth rate at all concentrations tested. A similar pattern could be observed for other strains as well, with a visible delay in growth rate as the concentration of potassium bichromate increased. Macroscopically, we have observed that in the case of 30% of strains, an increase in the concentration of potassium bichromate determined an increase in the production of pigment and secondary metabolites as a defense mechanism, and ultimately the growth of a less dense colony. In the case of 4 strains, no significant differences could be observed apart from a reduction in growth rate. In a small number of strains, low concentrations of potassium bichromate significantly and visibly stimulated growth, and an increase in concentration determined a significant decrease in growth up to a point of complete inhibition at higher concentrations. Microscopically, we have identified the presence of spores even at the highest concentration of potassium bichromate in the majority of strains. The exception was represented by 20% of strains for which complete inhibition of sporulation was identified at the highest concentration of potassium bichromate. In conclusion, we have confirmed the high level of chromium pollution in the study area, as well as the presence of stress exerted on the soil microbiome, but based on the high values of the microbial biomass and low metabolic quotient, we have supported the premise that metal-tolerant microorganisms might be present in the contaminated soil. In that sense, we have successfully isolated 19 filamentous fungi belonging to genera that have been proven through studies to have bioremediation potential in heavy metal contaminated soils, such as Trichoderma, Fusarium, Aspergillus, and Penicillium. Out of these strains, 3 of them were moderately tolerant to concentrations above 500 milligrams per liter of potassium bichromate, and 1 of them was very highly tolerant to all concentration tested. Lower concentrations of potassium bichromate stimulated growth, while an increase in concentration determined a delay or even complete inhibition in growth and sporulation. Future studies will focus on testing the tolerance of the chromium-resistant fungi to other heavy metals, as well as their potential to reduce the concentration of heavy metals in soil solution, and their potential to treat those heavy metal contaminated soils through combined bioremediation methods. This is the bibliography used for this presentation. We wish to thank the Ministry of Research, Innovation and Digitalization, and I also wish to personally express my gratitude to my PhD coordinators, my tutors, and my colleagues. Thank you for your attention.
Thank you for your nice presentation. If there are some questions in room or in the online, please, Mrs. Don.
Congratulations. So this is a part of your PhD thesis.
Yes.
Okay, I have seen the perspective, but do you intend to use that strain very tolerant to high concentration for bioremediation solutions to go a little more to applications?
Yes, we plan on extending the experiments in the field,
Mhm.
because that is very important in research. Usually extending the experiments in the lab, we have some results, and extending
Exactly.
them in the field is quite different.
But
so
you
we
need
plan on
you need to think that you need at least 1 year to do these studies on the field. So keep in mind this.
yes.
Okay. Good luck.
Thank you very much.
If there are some other questions in online, if no, thank you, Christina. We pass through the next presentation of Mr. Usman Farouk, biodiesel production from waste cooking oil using aluminum pillared clay as a catalyst. Mr. Farouk? Mr. Farouk, are you online in the room or not?
Can you hear me? Can you see my presentation?
Now it's fine, please. Welcome.
Okay. My name is Farouk Usman from the Department of Pure and Applied Chemistry, Federal University, Birnin Kebbi, Nigeria. The topic I'm going to talk about is the biodiesel production from waste cooking oil using aluminium pellet clay as a catalyst. We know that the fossil energy we are using now are associated with many problems, such as the issue of global warming, rise in oil prices, diminishing of the reserves. So that is why an alternative energy source must be exploited. And one of these alternative energies that can be exploited in Nigeria, especially, is the biodiesel, because we know it has some certain advantages over the petroleum diesel. But the problem of the biodiesel is the cost of production. That is why we are looking at the use of waste cooking oil as the feedstock for the production, so that it will reduce the costs. Just like also any other process, catalyst is very important. And in transesterification, the catalysts commonly used now are homogeneous, which are associated with problems also difficult to recover. They promote saponification, which is unwanted. So that is why we try to see if we can look at clays. And the clays, the one that interests us is the bentonite clay, which belong to the smectite group, and the montmorillonite, which is the major constituents of the bentonite. They are heterogeneous acid catalysts. They are found to be active in many catalytic processes. They are ideal green catalytic material because they are abundant. We have an abundant deposit of this bentonite here in Nigeria, which we can use. It can be easily processed. It can be separated from the product when done. It is non-corrosive. But all these properties, the catalytic activity of this bentonite can be improved by what we call the pillaring process. In this pillaring process, the interlayer, the aluminosilicate layer, the two aluminosilicate layer are separated. In between them, we have interlayer cations like sodium and calcium. So we can introduce aluminum or chromium to replace those sodium and calcium. And after that, it will be intercalated, dried, and then calcined. This new oxides that we introduce into the bentonite layer will form a robust oxide, and it will form a pillar. By doing this, it is going to increase the surface area, the pore volume, and sometimes even the pore size. So my materials here, the bentonite and the other Chemicals were purchased from Sigma Aldrich. The waste cooking oil was sourced from various restaurants here in Birning Kebbi. And then the process of the pillaring. First, we have to prepare the pillaring solution, which is the mixture of 0.25 molar of aluminum chloride plus 1 molar sodium hydroxide in the ratio of 2:1. After that, we mix it with 5% clay suspension. We put it in ultrasonic bath. We use ultrasonic bath so that it will save time for us. If not, we are going to use almost 24 hours to do that. Then the product will be washed with water until there is no chloride ion. You can test that by silver nitrate test, and then dry it 60 degrees, calcined 500 degrees for 5 hours. Then the catalyst produced is characterized using XRF, BET, and FTIR. Also, the biodiesel produced will be characterized using FTIR. Then transesterification of the waste cooking oil. Before the transesterification, we have to reduce the acid value through esterification. We have to remove all the solid particles there before we give we set to the transesterification at 65 degrees. Catalyst loading of 0.5 for 2 hours, and the methanol to oil ratio of 9:1. So this is the result of the XRF result. Yeah. So this is the XRF result. We are seeing, yeah, it's shown there. You see the aluminium oxide have increased because of the introduction of the aluminium, and it forms a robust pillar. Then the silica also reduced due to the overall percentage. It will rearrange the iron, magnesium oxide, the calcium oxide, and the potassium oxide all reduce. So this we can say that the pillaring has taken place. But to confirm that, that is why we use the BET. If you look at our raw bentonite, it has a surface area of 202.10, but after it was now escalated to 336, and then the pore volume from 0.125 have increased to 0.189, while the pore diameter, there is a slight reduction in there. So it's very important, this pillaring, because not only biodiesel, you can increase the surface area, the pore volume for other catalytic processes. So here is showing the FTIR of the pillared clay. We have from at 700 we have the formation of LaMgOH. At 907 we have the formation of LaOH. We have the stretching of SiO at 997, while at 1627 is the formation of OH of water, and then at 3400 is also stretching of the OH of water. But around 3400, when we compare it to the FTIR of the raw bentonite, there is increase in width of the peak of the OH of water because of the distillation and the addition of the aluminium there. Then we have the FTIR of the biodiesel, of which the spectrum region from 1500 to 900 elaborates on the chemical distinction between the biodiesel and the waste cooking oil, and then the presence of carbon to oxygen double bond stretching vibrations of carbonyl groups in esters causes the appearance of a strong peak at 1742. And then the peaks at 2922 at 2851 shows symmetric and asymmetric vibrations of carbon to hydrogen in CH2 and also in CH3 groups. This is actually an ongoing research. We have to use GCMS to see the fatty acids methyl esters, and also we have to test the petroleum properties of the produced biodiesel, and also compare this production to using raw bentonite to see the difference, or in terms of yield. The yield we obtained in the research is around 71%. So in conclusion, the aluminum pellet clay was successfully prepared and characterized. The study, which is part of an ongoing research, confirms the effective use of aluminum pellet clay as catalyst for converting waste cooking oil to biodiesel. The catalyst gave a biodiesel yield of 71%. Although the process requires further optimization, this can kickstart cost-effective biodiesel production process. Thank you for listening.
We thank you for your interesting presentation. If are some questions from online, Mrs. Doni, please.
Hello, congratulations for your work, and you are very welcome to our event. My question is regarding the ratio between methanol and oil is not too high. The concentrate, the number of parts of methanol, 9:1. I think that is not so advantageous to use so high concentration of methanol from economical point of view. What do you think?
Yes, I agree with you. If we come to the optimization process, we are going to reduce, or we are going to check 6:1. 6:1 is optimum in many researches, but we use this one so that we can have enough of it. For the end of the whole research work, we have to see, look at the methanol to oil ratio also.
Okay, thank you very much and good luck.
All right, thank you.
Okay, we invite Miss Anna Maria Baroi for the next presentation, exploring the possibilities of obtaining natural extracts abundant in phytoconstituents from grapevine wastes. Please, Anna.
Can you hear me? Can you hear me?
Yes, we hear you. We
okay.
wait the presentation
Okay, all right.
because we cannot see it. Can you share the screen? Anda, we invite you to share the screen because we don't see your presentation.
Can you see my presentation now?
Thank you.
Okay. So good afternoon. I'm Anna Maria Baroi, scientific researcher and member of team number 7, Emerging Nanotechnologies at NeXT-Chem, Bucharest. Today I will talk about natural extracts abundant in phytoconstituents from grapevine wastes. Cultivation of the vine, along with the process of wine production, are two traditional practices that have a beneficial effect on the socio-economic sector in various regions around the world. Grapevine crops are spread all over the world, mainly for grape production, which is divided into three main categories, like vine grapes, stable grapes, and dry grapes. Waste products are generated at each stage of winemaking process. And there are two distinct categories that show the origin of winery waste, those that results from the collection of grapes and those that results from the winemaking process itself. So the waste broadly involves grape stalks, grape pomace, along with grape seeds and wine yeast. For example, pomace represent about 20–25% of the initial weight of grapes, being the heaviest solid residue resulting from the processes of fermentation and pressing of the grapes. At the same time, in order to obtain the best quality grapes, different management techniques are applied to vineyards. Pruning plays an important role in the quality development of the vine and implicitly in the production of grapes, being performed in both cold and warm season. The pruning practice of vineyards in winter is the main source of residues, consisting in the accumulation of large amounts of vine shoots and canes. Considering the global area under grape cultivation, the estimated total volume of viticultural waste varies between 7 to 14 million tons. Besides the fact that these wastes are produced in a very short time worldwide, their accumulation can cause economic and environmental issues due to their organic matter, acidic pH, and salinity. These wastes have a low economic value and very limited uses. And the most common techniques for eliminating them consist in storing them on the field, and eventually they can determine water pollution, oxygen consumption in the soil and groundwater, as well as attraction of vectors of spread disease. In addition, compared to all aspects mentioned, from vine shoots to grape pomace or seeds, all the wastes are rich sources of bioactive compounds with beneficial effect for human health. The most abundant polyphenols present in grapevine varieties, according to literature, are represented by hydroxycyanamic acids, flavonols, stilbenes, and flavan-3-ols. So the aim of the current study was to obtain different extracts from grapevine waste with increased yield of phenolic compounds. The raw materials selected for this work were represented by two types of pomace, Muscato Tonel and Fetească Neagră, and other two types of shoots, Fetească Neagră and Tămioasă Românească. To extract the bioactive compounds from vine shoots and pomace, the raw materials have been shredded, and three solid liquid extraction pathways have been approached, namely one classical extraction using an oven, microwave assisted extraction with a microwave digestion system, and ultrasound assisted extraction using an ultrasound bath. In order to obtain an increased total phenolic content, two solid liquid ratios were studied, 1:10 and 2:10 weight per volume. So in the end, 24 extracts were obtained. Furthermore, to evaluate the total phenolic content of extracts, the Follin-Cioculteu spectrophotometric method was chosen. It can be observed the variation of the total phenolic content depending on the mass ratio used, the type of vegetal material, and extraction methods applied. By increasing the ratio of plant material to solvent, a higher phenolic content was achieved in all three cases of extraction methods. By comparing the pomace samples of Fetească Neagră and Muscato Tonel, the best results were obtained mainly for Muscato Tonel, showing the highest levels of the total phenolic content in all cases. In contrast, for Fetească Neagră and Tămioasă Românească shoot samples, the highest concentrations of phenolic compounds were reported for Fetească Neagră extracts, which were obtained through microwave and ultrasound assisted extraction methods. In conclusion, the concentration of phytoconstituents from waste strongly depends on several factors, including the used variety and type of waste, the ratio between vegetal material and the solvent extraction, and also the extraction methods applied. The future works include the use of these extracts rich in polyphenols for green synthesis of metallic nanoparticles. So these nanoparticles will be further characterized by different analytical and microscopic techniques, and the optimization of nanoparticle synthesis methods will also be carried out in order to obtain nanoparticles with desired size and morphologies. Finally, the nanoparticles will be evaluated to demonstrate their antioxidant and antimicrobial activity. Thank you very much.
Thank you for your nice presentation, Anda. I would like to invite to the questions, if there are some questions from online. Anda, I would like to ask you something about the stability of such type of nanoparticles obtained through photosynthesis. What are you expecting? How are you going to stabilize them?
It is expected that the more the phenolic content is higher, the more the nanoparticles can be formed in a spherical and in a smaller dimensions.
Yes, but the tendency is to agglomerate very fast. So a stabilization of them should be proceed.
I think the concentration of the metallic solution that is adding to the extracts is key in this point of view.
We will see. Thank you. Thank you
You're
very
welcome.
much. We invite Ms. Ioana Alexandra Bala for the next presentation, blue light laser effects on trichoderma on its plant biostimulant formulation. Please, Ioana.
Hello. Today I will talk about the blue light laser effects on trichoderma and on its plant biostimulant formulations. The environmental conditions like the level of oxygen, temperature, humidity, UV radiation, and the concentration of reactive oxygen species differ in soil and on the crust surface. These properties can change during day and night. Fungi developed the ability to adapt to ever-changing environment in order to survive. One important factor is light, which can regulate morphological and physiological processes of fungi and expression of some enzymes like the lignocellulosic ones. Light, especially in the blue regime, influences signaling pathways affecting the metabolism, stress response, and development of fungi. This study presents the influence of blue light laser radiation, a less used form of illumination, on the development and enzyme production by trichoderma. It also investigates a possible application of microalgae and microalgae trichoderma formulations as plant biostimulants. A cocktail from trichoderma enzymes combined with enzymes extracted from algae was tested on Arabidopsis thailandana seeds to observe the phenotypic morphology. Here I represented the strains that were used in our study. Fresh mature spores of trichoderma strains were inoculated into ISME or PDB medium. The developed mycelium from ISME was inoculated into water with rice husk, and the mycelium from PDB into minimal medium supplemented with whey and yeast extract, and they were incubated for 14 days at 28 Celsius degrees. The samples were exposed to blue light laser for 16 seconds at 1 laser intensity after 6 days of incubation. Erlenmeyer flasks were kept into the dark throughout the experiment. The cellulase, alpha amylase, and protease activities, lipid peroxidation, and sporulation of trichoderma were analyzed. The degradation of rye husk was visualized by SEM, and SPSS software was used for statistical analysis. After the determination of protease activity, the samples grown in the presence of RH after 7 days from irradiation, a more pronounced increase in activity was again seen, being significantly higher in irradiated samples for all strains. The best activities were observed for Trichoderma pseudoconingi, followed by Atrobruneum, Harzianum, and Rezei. These values are generally higher than at 28 hours. After 7 days, laser light treatment had a statistically positive influence on a proteolytic enzyme synthesis for Trichoderma harzianum strain. In the case of samples grown in the presence of whey as a substrate, ANOVA analysis shows that there are no statistical differences between the samples, as you can see in figure 2A and B, and there are some differences in the amount of proteolytic enzyme synthesized, but we cannot say that laser had a positive effect on their production. After 7 days from irradiation, an increase in cellular synthesis can be observed only for the irradiated Trichoderma strain using rice husk as substrate. It was found that carboxymethyl cellulose method is not very suitable for whey cultures, possibly resulting in complexes between whey proteins and carboxymethyl cellulose, which give high values in controls. Other methods will try to determine the cellulose activities. After 7 days from irradiation, in the case of samples grown using RH as substrate, laser light treatments have statistically positive influence on alpha amylase synthesis for Trichoderma harzianum, followed by Trichoderma pseudoconingi, and some influences can also be observed for Trichoderma rezei. In the case of samples grown in the presence of whey, after 7 days from irradiation, the treatment with blue light had a statistically positive influence on enzyme synthesis for Trichoderma coningi, as you can see in figure 5B, and marginally significant for Trichoderma atrobruneum. A conclusion, we can say that the irradiation influenced the enzymatic activities, either increasing or decreasing them, depending on Trichoderma strain, substrate, and sampling time. As you can see in figure 6, the lipid peroxidation of Trichoderma upon radiation is not significantly different than the control. Here we can see the analysis of conidia of the samples growth in the presence of rice husk. The irradiated versus non-irradiated differences on Trichoderma pseudoconingi, Atrobruneum, and Rezei conidia number are statistically significant, irradiation inducing an increased significance. In the case of irradiated and non-irradiated Trichoderma harzianum strain, the differences are marginally significant. In the absence of another method for quantifying chlamydospores, an image analysis method was improvised in order to determine the influence of the blue laser light. A fresh slide preparation containing small amounts of non-irradiated and irradiated Trichoderma mycelium respectively, dehydrated with filter paper, and after that stained with lactophenol blue were made. Chlamydospores were counted by acquiring images along the entire edge of the mycelium. Here are the microscopic images for our strains. After processing the images in Fiji, intensity of each transformed image was measured using the program to determine how much of the picture is mycelium, the black part. As you can see, certain calculations were made based on the intensity generated by the program. Here you can see that the number of chlamydospore is significantly higher in irradiated samples compared with the non-irradiated one. This activity aimed to monitor the deconstruction of the lignocellulosic biomass represented by rye-husk by stem analysis and silica release. In these pictures, we can see that Trichoderma harzianum induces a significant destructuration into the lignocellulosic matrix with the removal of silica layer. Here we tried to quantify the soluble silica released during incubation, and we can see that irradiation had a positive and statistically significant effect for Trichoderma harzianum. Here we try to determine the bioactivity of irradiated and non-irradiated Trichoderma strains on Arabidopsis thailand. The supernatant from our cultures was used to lyse microalgae culture and was tested as non-lysed and lysed formulation on the development and salt tolerance of A. thailand. These treatments were used for vernalization and germination of Arabidopsis thailand seed. The root length was determined using image G before the treatment with salt. In this figure, we can see that the inhibition of fruit growth, we can see an inhibition of fruit growth in length, and we can see also the secondary root development after the applied treatments, and no statistically significant differences was observed between irradiated and non-irradiated ones. After the germination, we treated the seedlings with these variants. And after that, we acquired some images, and then they were processed using Fiji to measure the rosette area. Here we can see the phenotypic development of plant rosette of Arabidopsis thailand using different media treated with distillated water with 50 millimolar sodium chloride and 100 millimolar sodium chloride. In absence of salt, the treatments show no statistically significant effect compared to the control, treatment 1, with 50 millimolars sodium chloride. The treatments 2, 3, and 4 are marginally significant positive. Compared to the control, and especially the treatments 3 or 4, we can see that the area of rosette is larger. In the case of the treatment with sodium chloride with 100 millimolar concentration, a marginally significant positive effect is maintained only in the treatment 2 and partially in treatment 3. And in treatment 4, we can see that the area rosette decreases to the control level. Here are the conclusions. Blue laser light modulates the enzymatic activities of Trichoderma in a complex, apparently hormetic manner. The irradiation does not induce significant lipid peroxidation and intensifies sporulation of Trichoderma. And microalgae with microalgae formulation act as plant biostimulants on Arabidopsis thailand under salt stress, but more in-depth studies are needed to maximize the potential of Trichoderma culture. And thank you for your attention.
Thank you, Ioana. A complex study and a nice presentation. Please, if there are some questions.
Hello, it's Nikki
In
speaking
online.
online now. Very good.
Mr. Varachio, I suppose, but we cannot hear you.
You don't hear me? Now you hear me?
Yes, please.
Okay, thank you. Very good work, very nice explanation. I have only a small question. Do you use statistics? That is very good, but you use only that graphical representation that is good, or other methods to compare different treatments?
No. Graphical representation. I use one way ANOVA, the Turkish and
Okay.
Bonferroni
And very good, because you said about statistical significance, but what about practical significance? What I like to mean, for instance, you could find a statistical significant, it's 1 millimeter difference, let's say, but maybe for one specific application that is statistical significance, maybe we need to be at least 7 millimeters for a practical application. It means it's very good, your step, but you also took into account something for application regarding practical significance after statistical significance.
We see some statistical differences, but in what experiment?
Maybe I could repeat my question.
Tell you.
You could
Oh,
find.
if I may, I think Mr. Varaki wants to say, is there a practical significance of the results of the differences, or only statistical, only mathematical?
No, practical.
I could repeat the example. You find statistical, it's 1 millimeter difference for a lens.
No, more.
No, just a second to finish. But for a specific application, practical, the direct application, you need at least 10 millimeters to be the difference. It means it's statistical different, but not practical significant.
Do you hear me? Sorry to interfere. Do you hear me? Do you hear me?
Yes.
Maybe you can discuss.
We are
this
here on Tolerate Free.
But we have to pass through the next presentation.
Yeah. Okay.
Preliminary studies on microalga growth enhancement by mimetic strigolactones. Mrs. Daria Popa.
Yes, hello.
Welcome, and please.
Thank you. Okay. Hello, my name is Daria Popa, and today I will be presenting you preliminary studies regarding microalgae growth enhancement by mimetic strigolactones. Microalgae are known for their high value content of active ingredients such as protein, fatty acids, pigments, and carbohydrates. Nowadays, microalgae are broadly used for various applications, including treating polluting waters, developing pharmaceutical supplements, producing biofuel or biogas, and last but not least, used as biostimulants for enhancing crop quality. So enhancing microalgae biomass is necessary, and even more adding value to it through increased metabolite production. Few substances are known to be stimulatory for microalgae growth. Strigolactones, being signaling molecules and also phytohormones, play an important role in plant response to biotic and abiotic stress. In this study, we focused on finding potential stimulatory effects on microalgae growth by mimetic strigolactones. Regarding materials, first, Desmodesmus communis and Chlorella sorokiniana, both freshwater species. In order to assess the biological effects of mimetic strigolactones, we tested 4 newly developed mimetic strigolactones by our partners, Emprosoftec, meaning SLF1, SLF2, SLF3, and SLF5. The final concentration tested was 10 to minus 7 molar concentration, which was gained by intermediate dilutions made with BG11 medium after solubilization chemicals in DMSO. The microalgae were cultured in autoclave BG11 medium under control conditions of light of 130 microeinstein, a temperature of 25 Celsius degrees, and 150 rotations per minute orbital agitation in an algatron growth chamber. First, when considering microalgae growth, optical density measured by reading the absorbance at 750 nanometers is the main growth parameter to be evaluated. To determine microalgae's pigment load, we first centrifuge each sample for 3 minutes, discharged to supernatant, and added on the microalgae pellets 2 milliliters of 100 Celsius degrees, not 100, sorry, 60 Celsius degrees preheated DMSO for extracting pigments. After 10 minutes of vortexing the mixture, we centrifuge it again and read the absorbance of supernatants. For measuring the chlorophyll fluorescence, we use the fluorometer that measures the quantum yield, which is represented by the ratio between PV and FM for light-adapted microalgae cultures, being the main indicator of functioning of system 2. To measure biomass, we added the remaining microalgae cultures in a pre-weighted Falcon tube and centrifuge for 5 minutes at 7,500 rotations per minute. Then the pellet left, we dried it at a constant weight in an electric oven at 60 Celsius degrees. Regarding results, optical density was measured once every 2 days. After 40 days, after 5 days of culturing Chlorella sorokiniana, we could observe cell growth increasement in 2 treatments comparing to control. From day 5 to the end of the study, the same trend was observed for treatment number 3, which was the strigolactone SLF3. In this case, a significant increase of sorbence was recorded comparing to control. At the same time, we could observe a stimulatory effect for Desmodesmus communis also by the treatment SLF1 and SLF5 this time. Regarding pigments, the extraction was made after 40 days of culturing, following the steps I mentioned before. And once we had the extracts, their absorbance was measured at 3 different wavelengths. Needs to calculate the chlorophyll A, chlorophyll B, total carotenoids, and summarize resulting in total chlorophyll. The total chlorophyll content was increased in chlorella supplemented with SLF3 by 66% comparing to untreated controls. And an important part being chlorophyll A. On the other hand, Desmodesmus communis pigment extracts were just slightly increased above control values for treatment with SLF3 and SLF5. Chlorophyll fluorescence is an indicator of photosynthetic energy conversion in microalgae. Measuring after 14 days of culturing, results pointed towards the same SL mimetics, SLF3 and Chlorella sorokiniana for Chlorella sorokiniana and SLF1 for Desmodesmus communis. This indicates a significant improvement in functioning of system.
O să vină ăla și o să spună că n-are bare de eroare.
Okay, biomass production. After weighting the microalgae biomass obtained after 14 days of culturing, the highest amount recorded was in the case of Desmodesmus stimulated by mimetic SLF5, followed by SLF1, with almost 25% more comparing to biomass resulted from controls. At the same time, biomass production confirmed the affinity of chlorella for mimetic SLF1 and SLF3, which were treatment B1 and B3. In conclusion, preliminary studies showed that the microalgae growth can be enhanced by at least 20% compared to control by supplementing the culture media with certain mimetic strigolactones at a 10⁻⁷ molar concentration, depending on the microalgae species, while Chlorella sorokiniana was better stimulated by SLF3 regarding cell growth, chlorophyll content, and biomass production. Desmodesmus had an affinity for SLF1. The mechanism behind these results through which the mimetic stimulate microalgae growth needs further investigation in order to be detected or explained. But even so, after assessing this mimetic strigolactones, we can conclude that significant stimulatory effects were recorded over Chlorella sorokiniana microalgae development. Further studies with different strigolactones concerning concentrations are ongoing. These are the references used. This work was supported by Segment Project Biolignol. And thank you for your attention.
Thank you for your presentation. If there are questions in online? No? Thank you, Dania. We move further and invite Mr. Grigore Pshenovsky for the next talk, Catalytic Pyrolysis of Waste Biomass. Welcome.
Thank you for your introduction. So today I will present catalytic pyrolysis of waste biomass. So let's begin with the presentation. First of all, to begin my presentation, what is pyrolysis? Well, pyrolysis come from two separate words. Means pyro means fire and lysis means separating. In other words, pyrolysis is thermal decomposition of organic or inorganic materials in an inert atmosphere or with insufficient oxygen or absence of oxygen to cause partial oxidation. Another definition of pyrolysis is molecular breakdown of larger molecules into small molecules in presence of heat. And also pyrolysis, you can search as thermal cracking, thermolysis, depolymerization. In this image, you can see an industrial scale pyrolysis in which the biomass is fed into a reactor. During the process of pyrolysis, you can obtain three different phases. Ash, which is solid, liquid, and gas. An interesting part about the gas, it can be purified and also recycled back to the process. This also common that I used the same process in my experiments. The solid phase is called charcoal ash biochar, while the condensable liquid is variously referred as pyroligneous liquid, liquor, or pyrolysis oil. The gas is called producer gas, syngas, or wood gas. Factors influencing pyrolysis process is pyrolysis temperature, heating rate, moisture of biomass, supply of oxygen, or lack of oxygen. And also from the first two factors, we can choose which method of pyrolysis we can use, slow one or fast one. Now I will discuss about the experimental that I made. The materials that I used for my for the pyrolysis was oak sawdust, having a humidity of 8%, and type of wood was oak, in Romanian stejar. Catalysis that were used also was dolomite and zeolite. The interesting about the zeolite was the particle size. One is fine as powder, and here you can see a bigger particle. But in my experiments, it's not a big deal. I didn't saw any difference. It was made in plant experimental part. This may be affect more in industrial scale. As the pyrolysis equipment that we used, you can see in this sketch with the nitrogen cylinder reactor, the electric heating resistor, gas liquid, or let's say the 5 is cooler, the glass or tube where is collected the fraction oil, and also the gas is collected in the bag, or also can be used in a GC analysis. For my experimental methodology, I weighted oak sawdust that was fed into a reactor. The reactor was assembled. After the reactor was assembled, the installation was purged with nitrogen to eliminate the oxygen from the whole installation. After the everything was done, the installation heat or temperature was increased to 450 to 500 Celsius. After everything is heating up, the reaction begins of the pyrolysis. We obtain the pyrolysis oil, and also after the whole process of the pyrolysis ended, we obtain the ash or biochar. For the pyrolysis oil, we used a GCMS. For the biochar, we used a NOVA gas sorption analyzer, or let's say BET, for pore area. And as a result, we started with the slow pyrolysis, what it means and why we made the slow pyrolysis, what it means. In the slow pyrolysis, it is important to have a minimum heating rate around 10, maximal 10 Celsius per minute temperature. What is important in this slow pyrolysis to obtain as much as we can of biochar. It is important because in slow pyrolysis is primarily obtaining biochar and lower oil and gas. Where we can use this biochar, it's interesting as a fertilizer or as a filters. As a result, for the biochar, this is the biochar that we obtained. It's interesting that in specific surface area, with the increasing of temperature, the specific surface area also increase. What it means, with increasing the surface, we are increasing the pores, the diameter of the pores that can absorb the other compounds. After the slow pyrolysis, we try the fast pyrolysis. What it means? In the fast pyrolysis, it's important to obtain as much pyrolysis oil in a heating rate, at maximal heating rate. Let's say the maximal temperature, in all experiments, the maximal temperature that we obtained was around 500, in range of 450 to 500. We obtained in this control one, fast pyrolysis, obtaining the 37 pyrolysis oil. The future results, I will refer to these results. I will compare these results. These GCMS peaks obtained, and also here are results of the analysis of liquid phase. Here we can see acetic acid with furfural that are volatile compounds that can be condensed, but also the phenol groups refer to the lignin. Maybe some of you know that lignin is very stable compound and it's hard to decompose it. And this is very important in this pyrolysis to decompose these phenol groups. In other words, less percentage of the phenol groups. Using the dolomite catalysis, we obtained, compared with the control one, we obtained with a 3% more oil, with 3% more, obtaining in the conclusion 40% of pyrolysis oil. Here you can see this image as a normal liquid, but using dolomite catalysis, I'm sorry for the image, it's kind of weird, but you are already, you can see a more pure, more fluent liquid in the pyrolysis oil. Here also a GCMS analysis of the liquid fraction. And here, after using the dolomite, you can see an increasing of the volatile compounds and decreasing of phenol groups. What it means? Decreasing, we already tried to decompose lignin. That is kind of hard. The next catalyst that we used was zeolite. Compared also again from the control fast pyrolysis, we already boosted the heating rate by 30%, and also increasing the oil compared to the control. Here you can see the results. Using the zeolite, we decreased the phenol groups, also, which it means decomposing the lignin from the wood. And we came with the question, with the idea, what if we combine zeolite and dolomite? What will happen? And here we come. We obtained not only boosting the heating rate, but also increasing the pyrolysis oil. And also, if you see in the image, a more pure liquid with the phases, that is two phases. Thank you. 2 minutes. Okay, 2 minutes. That is more easy to extract compounds that we need. Here also a GCMS analysis, and using the mix of two catalyses, we decreased more and more phenol groups, also decreasing the lignin from the wood. In conclusion, in pyrolysis process can be used a variety of wastes, such as wood, plastic, vegetable waste, animal waste, and etc. In this process, the heating is done at 40 to 50 degrees Celsius, and in this process, free oxygen is completely absent. Methane, biochar, and bio-oil can be obtained in this process. And catalytic pyrolysis allows the selectivity of the designed product, liquid phase, in higher proportion than simple thermal pyrolysis. Using catalysis also lower energy consumption than normal pyrolysis by judicious choice of catalysts. A synergic effect on the yield in the liquid fraction can be obtained. Thank you.
Grigore, thank you for your interesting presentation and for your clear presentation. If there are some questions, Mrs. Doni, please.
Thank you.
I hope that you will patent this, no?
Patent? Not to patent, because these experiments are some of the articles. Many experiments were made but about the patent, we
but
can
the procedure,
use
the procedure of the mix.
This mix of the catalyst. It's a good idea. Thank you. It's an interesting one. Any?
Some other questions from online, if there are. If not, thank you, Grigore.
Thank you.
Yeah. And we invite Mrs. Victoria Brânzari for the genotoxicity assay as a screening tool of plant biostimulants, the case of fermented horsetail. Please.
Hello. Please. My name is Victoria Brânzari, and today I will present you about the genotoxicity assays as a screening tool of plant biostimulants, the case of fermented horsetail. The ongoing development of plant biostimulants requires not only tests of their stimulatory potential, but also tests to prove their safety on plants, as the line between the biostimulant effect and the toxic effect needs to be known. When we talk about safety in terms of genotoxicity, we mean lack of any genetic damage, as present of micronucleus or irregular prophase or ring formation and so on. Any deviation from the classical cell cycle, mitosis or cytokinesis, is considered cytotoxic and genotoxic. The potential toxic or biostimulant response of a product can be determined using the genotoxicity assay performed on Visea-Faba, known by all as Faba-Bin. This technique performed on eat-root meristems and comes with many advantages for, especially for industrialists, as it would hold arguments on safety of the delivered plant product. The aim of this study was to investigate the fermented horsetail as a possible plant biostimulant by determining the seed germination rate, root elongation inhibition, micronucleus frequency, and mitotic index. Horsetail is a plant known to be rich in silica and used as a plant supplement. In our study, we used fermented horsetail collected at different periods, like 1, 2, and 3 weeks. For the germination process, first Faba seeds were subjected to surface aseptization. For treatments, we propose to use a positive genotoxic control, like lead nitrate. After 5 days of germination under temperature-controlled conditions, the root tops were collected and fixed in ethanol with acetic acid. Then the material goes through a process of hydrolysis and staining to obtain a clear visualization of the cell division process. As far as the cellular part, seed germination rate and root elongation inhibitions were first calculated to see if our product stimulated or inhibited the seed germination. Here, the 1-week maceration sample induced better rate germination, like 93% compared to control, who contain sterile distilled water. The same sample gave the best results following the Ray index. The roots showing slower growth on 2 and 3 weeks maceration. Mitotic index results show an intense cell division in plants subjected to treatment with fermented for 2 weeks, followed by the same sample, in addition with lead nitrate, compared to control. Here we have in this image, it's an image from a negative control with distilled water, and it's a normal mitosis. The micronucleus assay has become one of the most popular methods to assess genotoxicity at different chemical and physical factors that not necessarily kills the cells, but only damage their genetic material. Analyzing the image above and this one, you can see a difference related to the presence of a very large number of micronuclei. These are small membrane bonding compartments with DNA content encapsulated by a nuclear envelope and specially separated from the primary nucleus. As be confirmed by the data obtained, the frequency of micronuclei in positive control containing lead nitrate far exceeds the result of horsetail fermentation on 1 week, 2 and 3 weeks. In the following images are represented the cell divisions from the horsetail fermented separately and in addition with lead nitrate. Micronuclei were also reported in some places, but not significantly present. The most interesting fact here is that an intense proliferation of chloroplast is
We cannot hear you.
Observed. An explanation in this way is now understood. And now? Do you hear me?
Now, yes, please continue.
According to European standards in the soil quality and assessment of genotoxic effects on higher plants, the 1 week horsetail maturation shows initially a slightly genotoxic effect, by after 2-3 weeks of fermentation. This effect seems to disappear and the extract becomes slightly biostimulant. Even more, the presence of stress, the macerate shows protective action at the cellular level, agonist to genotoxic substance, as the micronucleus assay and the mitotic index results show. In conclusion, we can say that horsetail maceration successfully responded to the genotoxicity assay of VCA-FABA, an approach that can provide good tools in identifying possible genotoxic effects on plants, and an excellent screening of chemical products on plant biostimulants surely. Considering the lack of chromosomal aberration in the process of cell division and the lack of cytotoxicity, according to mitotic index and the frequency of micronuclei results, in combination with the protection against genotoxic abiotic stress, the horsetail maceration provides to be a promising product. Thank you for your attention.
Thank you for your presentation. If there are some questions from your colleagues or from online. If not, Luminica, please proceed with the next presentation, the enhancement of the biological activity of the honey and its biomimetic death with polyphenols from honeysuckle, Linocera caprifolium flowers.
Hello, everyone. My presentation for today is about enhancing the biological activity of honey and its biomimetic antibiotic solvent with the polyphenols from honeysuckle flowers. Honey is a natural product obtained by honeybees and with several biological activity. Also, honey is considered to have characteristic of natural deep eutectic solvent due to the intermolecular interaction between monosaccharides and disaccharides, and hydrogen bonds formed between them. In this study, we propose to enhance the biological activity of honey by increasing the antioxidant, prebiotic, antibacterial properties. And to achieve this, we mixed honey with dried honeysuckle extract after solvent evaporation. Also, we prepared a deep eutectic solvent which mimics honey based on content of main sugars found in honey, like which is glucose, fructose, and sucrose. And another objective of our research was to demonstrate the similar behavior between the honey and deep eutectic solvent. To achieve this, we follow the same protocols used for honey. We prepare the mixture of this with honeysuckle extract. First of all, we have characterized the ethanolic extract of honeysuckle flowers by determination of total polyphenol content, total flavonoid content, and total hydrocinamic acid. Also, we phenolic acid and flavonoids were analyzed by HPLC. The antioxidant activity of the sample were analyzed for spectrophotometric methods, FRAP, CUPRAC, DPPH, ABTS assays, and the interaction between honey and honeysuckle extract were analyzed by determining a combination of index, and the lowest value of combination index we can observe in the case of FRAP, honey and a mixture of honey and honeysuckle extract has a combination index less than 1, which means a synergistic behavior, and we can say we can see that this honeysuckle extract mixture was the same behavior. And this slide we are presented is a hologram of our product, and this hologram is a graphical representation of interaction between two or more compounds. And our case is about honey and honeysuckle extract, or this and honeysuckle extract. Also, is a program is a plot used to compare the effect of individual compound with their combined effects when are used together. On this slide, we can observe that the similarity of hologram and the combination index in the case of FRAP for mixture of honey and honeysuckle extract. Also, we can say here there is a hologram of our product by in the case of IBTS and DPPH assays. Well, we can observe that mixture of honey and extract has additive behavior in the case of IBTS, and in the case of DPH, observe the antagonistic behavior. In this slide, we can observe the concentration dependence of experimental and theoretical antioxidant activity by FRAP and the Kuprak methods. The antioxidant activity varied linearly in the case of FRAP and Kuprak methods, and sigmoidal in the case of DPPH and the BTS method. Also, we can observe that experimental value of mixture of honey and extract are higher than theoretical value. Okay. The antibacterial method was, as I by determination used the agar disk diffusion method on gram-positive and gram-negative strain, and the mixture shows the has the antibacterial properties for the strain Bacillus cereus, Enterococcus fecalis, Enterococcus fecalis, Rhodococcus equi, Escherichia coli, Pseudomonas aeruginosa, Enterobacter aerogenes. Also, antibacterial activity was performed by determination of minimum inhibitory concentration, and we can say that mimic value of mixture in the case of Escherichia coli was 50 milligram per milliliter, and for Bacillus cereus 200 milligram per milliliter, and on the case of Rhodococcus equi, there are difference between the mixture of honey or and the mixture of dipiotetic solvent with the extract. Our concentration of our product tested exerted bacterial growth promoting on Lactobacillus reuteri compared with honey and the DS simple. We can observe that the highest activity, the prebiotic activity was at the concentration 25 milligram per milliliter of our products compared to honey and DS individually. In conclusion, we can say that during this research, we obtain a product with enhanced biological activity, and the interaction between honey and extract or DS and extract in the term of antioxidant activity range from synergies to additive and antagonist, and also the dipiotetic solvent which mimics with honey showed a similar behavior in most of cases. Thank you.
Thank you for your presentation. If there are some questions in the online, Luminita, I want to ask you something about the stability of your product after you add your compound. Stability in time of your final product. Have you performed some studies? Ongoing study? Because it's very important if you can improve the antimicrobial, antioxidant, and so on, the stability in time of such product will be interesting to be studied and to decide. Okay. Okay, thank you. We go for the next presentation, the last one, and we invite Eliza Gabriela Brettelfeld for CO2 capture using eggshells as ecological solvent from agricultural waste.
Hello, can you hear me?
Yes, welcome.
Thank you for the nice presentation. Just a second. Hello, my name is Eliza, and today I will be presenting you another CO2 capture method, namely CO2 adsorption using eggshell as ecological sorbent. The egg industry is a significant agricultural sector with high economic importance. Can you imagine food without eggs? Eggshells are considered in European Union a size stream and hazardous materials. Therefore, something must be done in order to protect the environment. Eggshells are primarily sourced from the egg industry, where they are generated as a byproduct of egg production. They are obtained from eggs laid by various species of birds, with chicken eggshells being the most commonly encountered. These eggshells are typically discarded as waste after the eggs are used for human consumption or processing. The challenges associated with the disposal of eggshells include their slow decomposition rate, occupying significant landfill space. They release also methane, a dangerous greenhouse gas contributing to the climate change. Eggshells can be recycled through various utilizations. Soil amendments or fertilizers due to its rich calcium composition, calcium supplements, animal feed additives, and even the creation of ecological, eco-friendly construction materials. Another use is the CO2 capture, the aim of this study. CO2 and culture looping is a carbon capture and storage technology that aims to reduce CO2 emission from the industrial processes. In this project, calcium oxide or limestone is used as a sorbent to capture CO2 from flue gases. And guess what? Eggshells are made almost entirely of calcium carbonate crystals. The captured CO2 reacts with the sorbent to form calcium carbonate, which can subsequently be heated to release pure CO2 for storage or utilization. Calcium looping offers potential advantages such as high capture efficiency, low energy penalties, and the ability to produce a concentrated CO2 stream. It is considered a promising technology for reducing greenhouse gas emission and mitigating the climate change. In this study, eggshells from hatch eggs were prepared as adsorbent materials for CO2 capture. They were washed with water, mechanically separated from the inner membrane by crushing, air-dried, grinded using a centrifugal mill, and then calcinated. Thus, calcium oxide for chemical looping was obtained. The following step was the transformation of anhydrous magnesium chloride into nano-magnesium oxide using distillated water and stage calcination, as we can see in the three reactions, releasing the hydrochloric acid in the system. The two powders, calcium oxide and magnesium oxide, were mixed together at 85% and 15% respectively, dissolved in water for best mixing, and recalcinated and milled to micrometric size particles. FTIR analysis revealed the high reactivity and hygroscopicity of calcium oxide through the traces of CO2 and water adsorbed. XRD confirms the 15% nano magnesium oxide content as intended before, and the presence of calcium oxide, calcium carbonate, and calcium hydroxide. The nanoparticles of magnesium oxide has a size particle of 50 nanometers. The capacity to capture CO2 was determined using a thermogravimetric analyzer from TA instruments with two purging gases, with 14% of CO2 and the rest of it nitrogen. The calcium oxide and magnesium oxide mixture showed more than 3 times higher activity towards CO2 than the simple calcium oxide granules after calcination in air. Nanomagnesium increases the CO2 capture capacity of eggshells. Together with calcinated eggshells, calcium oxide, magnesium in the form of nano helps to avoid the formation of a preferential gas flow path through the sorbent mass. The future perspective of eggshells encompasses various exciting possibilities in different domains. Here are some potential areas of development. Overall, the future perspective of eggshells revolve around their utilization as sustainable materials, their contribution to agriculture and horticulture, waste management, medical application, environmental remediation, animal feed and nutrition. These developments align to the principle of sustainability, resource efficiency and circular economy, opening up new opportunities for the beneficial of utilization of eggshells. Thank you for your attention.
Thank you for your presentation. If there are some questions from online. If no, I would like to thank for this and to thank to all the participants for the nice presentation from today. I will give the last word to Mrs. Doni to close the session.
Thank you very much. So we are the end of the first day. We thank you all for staying to this hour, and also from the people from online, because I have seen that there are around 40-50 attendants in online. So thank you very much, and see you tomorrow morning at 9:30 in the second day. We will have one invited lecture, a keynote lecture, and then the other presentation of the young researchers. And also please participate to the roundtable at 15:30. It will be a very interesting roundtable with our colleagues which are now abroad with research stages, and we hope that these colleagues will tell us how and yes, how they are evolving there, and which are the challenges in other laboratories abroad. So please, I hope that will be an interesting debate tomorrow afternoon, and then we'll have also some diplomas to share with you, and some little presents for for the best presentation. So have a nice afternoon today, and see you tomorrow. Thank you.
Thank you. Thank you.